Christian Albrecht University of Kiel in Germany also. And then after completion of his habilitation, or something like qualification for permanent faculty position at the German universities in 2006, he accepted a faculty position at the University of Michigan, and where he started in September 2006 as an assistant professor. And he received a number of awards, including a GSPS, it is from Japan, I think, yeah? And SF Career Award in 2009, and the 3M Non-Tenure Faculty Award 2011, yeah, and also the prestigious John Dewey Teaching Award, yeah, from the USA, I think, and the Harold
from Michigan,
Johnson Diversity Service Award from University of Michigan in 2018, and from 2007 until 2011, he was the Corning Assistant Professor of Chemistry, and in 2012, yeah, he was promoted to Associate Professor with tenure, and in 2016 to the rank of Professor. It's like a full professor. And his work is focused on the coordination chemistry of nitric oxide and its derivative as its partner to biological system, especially for nitric oxide sensing and detoxification. And more recently, he has also developed a research program in biocatalysis, like artificial metal engine and electrocatalysis. The latter being focused on immobilization molecular catalyst on electrode surface. This is suitable with Professor Fitri, I think, the field, yeah. And to derive the energy-related reaction, like especially proton reduction, yeah. And particular expertise of his group is the application of physical and theoretical method to coordination compound and corresponding catalysts. And if we check in the Scopus, his index his H index is around 42. Yeah, it's very high. It's very senior professor here in the US. And not too long here, I will give the time and the stage here, stage in Zoom, I think, to Professor Lehnert to deliver the lecture. Please, Professor Lehnert, now is your time.
Okay, thank you very much for the nice introduction. Let me share my screen. Let's see if that works. All right, can you see my screen?
Yes. Yes.
Okay, good. Then let's see if I can activate my laser pointer here. All right. So yeah, thanks again for the kind introduction and for inviting me to give an online lecture to your students and talk about nitric oxide, which is one of my favorite topics in bioinorganic chemistry. So here's a quick overview of my presentation. So I will first tell you a little bit about the history, you know, the research of nitric oxide and how it became apparent that it is relevant in human health and in biology in general. And then I want to focus on two topics that are very important that I want your students to learn about. The first one is how actually nitric oxide is made in the human body, right? How it is biosynthesized. And then the second one is once we have it made, nitric oxide is used for signaling in the human body. So how is the signal transduction working? And especially, you know, if you have a signaling event, you have a molecule that's generated, that's released, and then you need a sensor, right, that binds that molecule and then generates a signal. So how does that work? And the key enzyme here is called soluble guanylate cyclase, which is the human NO sensor protein. So I want to talk about how that works. And this is also a good point then, if we have time, I can talk a little bit about our research where we're also looking at how, you know, how this activation process works on a molecular level. But if we're out of time, I can also skip that. And then finally, I have one slide to conclude my presentation. Okay, so the first question is, what does a bioinorganic chemist do with nitric oxide? Right, nitric oxide before about 1980 was mostly considered to be an air pollutant. It's produced in the burning of fossil fuel, and it's a important component of smog. So, you know, when you see the pictures with the brown haze, you know, over a city, the smog, and then that is NO2 that comes from nitric oxide NO when it reacts with oxygen. So NO is a molecule with a bad reputation, right? Is what I sometimes say. And of course, we use nitric oxide gas in my lab. And so if you look at the gas cylinder, right, you can see nitric oxide here is classified as very toxic, corrosive, oxidizing, and it's even dangerous for the environment, right? So you might wonder, you know, what does NO have to do with biology, right? And all of this really changed about around the 1980s, and it was due to the work of especially these three gentlemen here, who were rewarded with a Nobel Prize for medicine in 1998. And so the story is that it was known for quite some time that our body in the endothelial cells, so these are the cells that line the inside of your arteries, these cells produce a molecule which was called the endothelium-derived relaxing factor that induces vasodilation, right? So it widens your arteries and it lowers the blood pressure. But it was not known what this molecule is, right? And so they figured it out, right? That the molecule that is actually responsible for this signaling is nitric oxide. And that was pretty surprising, right? Because I told you about the history of nitric oxide. It's a very toxic molecule. And so it was a big surprise when it was found that nitric oxide is a signaling molecule in the human body, right? And that was such a profound discovery that it was actually awarded with a Nobel Prize for medicine. So here are some of the things that nitric oxide can do. And I want to stick to especially the cardiovascular system, because this is something that we all know about. We know about blood pressure. So this is a good example that we can relate to, right? So here is a slice through an artery. And you can see here outside in red, or here in the zoom, this is this smooth muscle tissue that surrounds the artery. And then here in blue, these are the endothelial cells. And the endothelial cells now contain an enzyme that produces nitric oxide, right? And that nitric oxide diffuses into the smooth muscle tissue, where it's detected by soluble guanylate cyclase. And then when nitric oxide is detected here, it induces relaxation of the smooth muscle tissue. And when the muscle tissue relaxes, then your artery becomes wider, right? And this is what this graphic shows. So going from lower to higher relaxation, you can see smaller diameter, larger diameter, right? So the blast vessel relaxes, it widens, it dilates, and the blood pressure drops. And so this, you know, the state of dilation of your artery is directly related to the flux of nitric oxide that comes from the endothelial cells and goes into the smooth muscle tissue. Now, nitric oxide is very toxic, but our body has found a way to deal with that. And the secret is concentration, right? So all this signaling of NO happens at these about nanomolar concentrations, and at these concentrations, nitric oxide is not toxic. So that means, though, we have to have a very, very good sensor, right? We need to have some kind of protein that can detect nitric oxide at nanomolar concentration. And I'll show you that, and I'll show you how soluble guanylate cyclase is doing that. So nitric oxide is produced in the cardiovascular system. It's actually also produced in neurons. It's produced in your brain and is involved in signal transduction in the brain. And then on top of this, which is really interesting, our body actually makes use of the fact that nitric oxide is toxic and also uses it for immune defense. So for example, your macrophages, which is shown in this cartoon here. So this orange blob here is a macrophage. Is attacking these little green E. coli cells, right? And one way to do it is it engulfs the cell and then it releases toxic chemicals. And one of the main immune defense agents that's used is nitric oxide. So our body makes use of the fact that this stuff is very toxic and uses it for immune defense. And in this case now, our body produces up to micromolar concentrations of NO, right? So that is 10 3 orders of magnitude higher concentration, and that's the threshold when it actually becomes toxic, right? And we can use it for immune defense. Okay, so we can say then that nitric oxide is actually a very important molecule for human health, but we want to use it either at low concentrations in signaling or at very, confined locations like the area of a macrophage when it's used for immune defense, so it does not get toxic to the human body. yeah, let's skip over this. So the first question then is, how is nitric oxide actually made, right, in the endothelial cells? And the answer is, there is a dedicated enzyme called nitric oxide synthase that is responsible for producing nitric oxide in the human body. And there are 3 isoforms of nitric oxide synthase, or NOS is the abbreviation for it, right, that are classified depending on location. So the endothelial NOS, or eNOS, is the enzyme in our endothelial cells, right, responsible for controlling blood pressure. Then we have the so-called neuronal NOS, or NNOS, which is found in neurons, and it's involved in signal transduction, and it's produced in the brain, as I mentioned. And then there is iNOS, which is inducible NOS, and this is the one used in immune defense. And so we'll show you in a second, these enzymes are relatives of cytochrome P450 family. So they catalyze oxidative chemistry to make NO, right? And this stuff I've already mentioned. Okay. So here is a very simple cartoon of NOS. So NOS is a homodimer, right? So the enzyme has 2 identical subunits, and you can see I color-coded this here. So one subunit is blue, and the other subunit is red, and this is the state where the enzyme is inactive. And in each subunit, we have 2 domains. We have a reductase domain. You can see there's some of the typical flavin cofactors in there that are used for electron transport. And then the key thing here is the oxygenase domain. This is where the chemistry happens, right? This is where nitric oxide is actually made in some oxidative process. So people have studied these enzymes for quite some time, and we have crystal structures, so we can take a closer look at what these, what these domains look like. So here is a crystal structure of the reductase domain. Here's the PDB where I took this from. So you can see here are the flavin cofactors, the FMN, the FAD, and here is the NADPH molecule, right? And this is how electrons are delivered from the outside into the reductase domain. So this will be this part here, right? And then we have the oxygenase domain. And in the oxygenase domain, we have a heme cofactor. So this is a heme enzyme. And you can see this heme has axial cysteine coordination here, right? So if you look at the active site here, it looks like a cytochrome P450, right? You can see in this crystal structure, the substrate is L-arginine. I'll get to this in a second. Is actually bound in the active site, and you can see that the guanidinium group here of L-arginine is right on top of the heme, about 4 angstroms away. And so we are getting ready here to do catalysis, right? So, but in this in this form, the enzyme is still ferric, so it's still oxidized, and the reaction cannot start. Okay, and then on top of this, we have this hydrobiopterin cofactor here, also in the active site. And this is different from cytochrome P450s. They don't have this type of cofactor, and it's right next to the heme, and we'll see it's also important. Okay, so the first important question is, how can we, or how does the body regulate the activity of nitric oxide synthase, right? Because there has to be a way for the body to turn it on and turn it off. If the enzyme would be always on, it will produce too much nitric oxide, it will kill us, right? And that's not good. So the body has to have a way to control how much nitric oxide is made. And this is indicated here by these white spheres. This is a binding domain for calmodulin, right? So calmodulin is a small protein that binds calcium, and it controls the activity of NOS. So when the calcium concentration in the cell goes up, the calcium binds to calmodulin, and calmodulin binds here, which activates the enzyme. And then if the calcium concentration goes down, the calmodulin falls back off, and the enzyme is turned off. So this is actually here, this is the inactive state of the enzyme. So you might ask yourself, why is this inactive, right? You have the reductase domain here, you have the oxygenase domain, the electrons, why don't the electrons flow from the reductase domain to the oxygenase domain? And the answer is, within one subunit here, they're too far apart. This distance is too large, so electrons cannot flow from the reductase domain to the oxygenase domain of the same subunit. Now, when calmodulin binds, something really interesting happens, right? When calmodulin binds here and here, there is a conformational change of the homodimer in a way that now the red oxygenase domain gets close to the blue reductase domain. So electrons flow from the blue reductase domain to the red oxygenase domain, right? And vice versa, here, electrons flow from the red reductase domain to the blue oxygenase domain. And so in this form now, the enzyme is turned on and nitric oxide is produced. And then again, when the calcium concentration goes down and calmodulin here is released, right, then we'll go back to the inactive state. So this is the way how the body can turn on and turn off the enzyme in order to control how much nitric oxide is made and make sure that the concentration is always kept at the low nanomolar range. Okay, so how is nitric oxide made? This is also interesting. Nitric oxide is made by metabolizing L-arginine, which is of course one of our 20 essential amino acids, right? And it's a two-step reaction. The first reaction is a hydroxylation reaction, which is shown here. So the guanidinium group here gets hydroxylated to this N-hydroxyl-guanidine. And then the second step is an oxidation reaction where we produce citrulline and nitric oxide is released from this group here. That becomes NO and it is released. So we have to run through two reactions here in order to ultimately make citrulline and then nitric oxide. So then we can take a look again at the active site, right? The cytochrome P450-like active site with the heme and axial thiolate coordination. And it turns out that indeed the reaction in NOS is actually very similar to cytochrome P450. So I don't know if the students have already learned about cytochrome P450, but I want to just quickly go over this. This is the standard mechanism of oxygen activation by cytochrome P450 enzymes, right? Where you start here in the resting state, which is the ferric state of the enzyme. You bind substrate, which is this RH here. In our case, it would be arginine, but of course, cytochrome P450s can work on all kinds of different substrates. So it's a general substrate here. And when the substrate binds in the pocket, the water molecule is pushed out and the heme becomes 5-coordinate. And that triggers electron transfer. So the heme gets reduced to iron 2. And once the heme is reduced to iron 2, it can bind oxygen. So you make your oxy intermediate here. This is the complex between iron 2 and O2, which is best described as an iron 3 with about no superoxide. And then there's more electron and proton transfer. You make the hydroperoxide intermediate. It's also called compound 0. And then this gets protonated here at the distal oxygen. The water gets cleaved off here by heterolytic cleavage of the OO bond, and we end up with the famous compound 1. Right? This is the famous oxidant that cytochrome P450s use, and how they were able to activate inert CH bonds. Even very strong CH bonds can be activated by cytochrome P450 through this very powerful oxidant, which is called compound 1. It is an iron 4 oxo species where the porphyrin ring is also oxidized by 1 electron, which is indicated by this radical sign here. So formally it's iron 5 oxo, but it's really an iron 4 oxo and an oxidized porphyrin ring. So this species will attack the substrate gets hydroxylated, and we return the cycle back around. So this is the canonical cytochrome P450 cycle, right, that people have studied in much detail. And it turns out that NOS uses a similar reaction to do its chemistry. So in the case of NOS, which is shown here, things actually evolve pretty similarly, but of course you have the tetrahydropterin cofactor also involved in the reaction. So this again is, go back, this cofactor here, right, which is redox active, and it's right next to the active site, and it can be used as another source of electrons in the reaction. So we look at the cycle though, right, it looks very similar. We start with the ferric form, it gets reduced to ferrous, oxygen binds to get us to this iron-free superoxide species, and then now electron transfer comes from the pterin cofactor, not externally, which makes a peroxyl species that gets protonated immediately. That gives us compound 0, right? The iron-free hydroperoxyl, which we protonate at the distal OH group here, cleave off water heterogeneously here, heterolytically, sorry. And then water is released and we end up with compound 1. And compound 1 is also the species now which is believed to catalyze the first step of the reaction, right? Of L-arginine hydroxylation, So the compound 1 will attack an NH bond in the guanidinium group of L-arginine and hydroxylate it, and that gets us to first intermediate. So we get back to our cycle. The first reaction here is catalyzed by compound 1, and we make the so-called NOHA, which is N-hydroxyl L-arginine. So this species is made, and then we need a second. We need to run through the cycle again. So we need 2 equivalents of oxygen, right? We run through the cycle twice in order then to oxidize now the NOHA here to citrulline. So interestingly, though, in the second step, so when we go back to this, in the first step, we run through the whole cytochrome P450-like cycle to make compound 1. But scientists believe that the second reaction, the oxidation reaction of NOHA, actually is catalyzed by either this peroxo or this hydroperoxo species. And this is shown here, where the peroxo hydroperoxo species attacks the guanidinium group like this. So this would be a nucleophilic attack, right? Here, making a oxygen carbon bond and then cleaving the O bond, ultimately then releasing NO and oxidizing the NOHA to citrulline and releasing NO. This is, of course, also an area that has been investigated for a long time. These reactions are very fast, so you can imagine it's not easy to study them, right? And so people have worked on this for a long time, trying to figure out how this works, but this is our best hypothesis now. So the first reaction goes through compound 1, the second reaction uses the peroxo intermediate to ultimately make NO. Okay, so here's a side view of the endothelium. So here's our blood, right? This is the inside of the artery. Here is an endothelial cell. Now in the side view, kind of sliced through it. And then here's the smooth muscle tissue right here, right? So I showed you now that in the endothelial cell, we have NOS, right? Endothelial NOS. And it takes up oxygen and arginine. It makes citrulline and nitric oxide. Now the nitric oxide can diffuse into the smooth muscle tissue. That's the important part here for us, right? It gets detected by soluble guanylate cyclase, and that induces relaxation of the smooth muscle tissue. But of course, diffusion is an unidirectional motion, right? So you can diffuse in any direction in space. So some of the nitric oxide will also diffuse into the blood, and it has important functions there as well, because nitric oxide also inhibits the attachment of platelets to the surface of the endothelium, right? And so it inhibits platelet aggregation and blood clot formation. So nitric oxide actually also keeps the surfaces of our arteries clean by inhibiting the aggregation of platelets, which is another important function also for our survival, right? Because if there would be uncontrolled platelet aggregation in the blood, we would all die of blood clots pretty early, and our lifetime would be very short. So nitric oxide is also important from that point of view. But what we, of course, today's lecture will focus on nitric oxide diffusion here into the smooth muscle tissue, and we're asking ourselves now how that signaling process works. So this is kind of quickly summarized here, right? So under other important functions of NO that are not related to controlling vasodilation is, as I mentioned, inhibition of platelet aggregation and blood clotting, inhibition of protein adhesion to the endothelial cell wall, and also NO, as we know, is an antimicrobial agent, so it also fights infection. Okay, but now the important question, NO sensing. So here's the picture again. This is very important, so there are many different ways to look at this. So here are, again, the endothelial cells, or an endothelial cell. Here's NOS, right? Oxidizing L-arginine to citrulline, making NO. Here's the cell wall, and this is the smooth muscle tissue. So NO diffuses across the membrane here, and here is our detector now. This is soluble guanylate cyclase. Oops, this is my clock. This is the enzyme responsible for NO detection. So you can see from my drawing that this is a heterodimer, so 2 different subunits, an alpha and a beta subunit. And then here is our, as a heme, that is used for NO sensing. And when this enzyme detects NO, it turns on and then it produces cyclic GMP. And I'll talk about all of these things in more detail in just a second. But this is the overall scheme, how NO sensing works in the endothelial cells in our blood, in our blood vessels, in our arteries, but also in the brain or in neurons. It works in a similar way, where in that case, then NOS generates NO, and then again, soluble guanylate cyclase is responsible for detecting NO, right? And so the signaling process in the brain, in the neurons, works in a very similar way as the signaling process here in our arteries. All right, so here's the domain structure of soluble guanylate cyclase. Again, we have two identical subunits, right? We have an alpha and a beta subunit, and the two subunits, they line up right next to each other, and then they intertwine with each other, right? So they run in parallel. You don't have, let's say, alpha subunit here, and then the beta subunit here, and they're kind of bound together. No, the two peptide strains are actually intertwined with each other. They run essentially in parallel, right? And this is shown here by the domain structure. So here's the alpha subunit, here's the beta subunit, and you can see that we have this so-called HNOx domain. This is the NO sensing domain. And you can see my little cartoon, here's a heme, right? By this little circle with iron in it, that's the NO detector. And then we have the PAS and the CC domain. These are the domains that connect the sensing domain, and this is the catalytic domain that actually catalyzes the production of cyclic GMP. And the important thing here, of course, is that the sensor domain here has to control the catalytic domain. So when NO is bound, the catalytic domain is turned on. And when NO is not bound, the catalytic domain is turned off, right? And so now you can see the NO concentration, right? That the flux of NO that enters the smooth muscle tissue then will determine the activity of SGC, soluble guanylate cyclase, and will determine how much cyclic GMP is produced in the catalytic domain. And that controls the level of relaxation or contraction of the smooth muscle tissue, and that controls the level of dilation of your artery. So soluble guanylate cyclase is a protein that is very difficult to work with. It is difficult to express it recombinantly. It is difficult because it's sensitive, it decomposes. And so scientists have not been able to obtain a crystal structure of soluble guanylate cyclase with the traditional method, right? Where you grow the crystal of the enzyme or the protein and then use X-ray diffraction to determine crystal structure. Has not been possible for full-length soluble guanylate cyclase. However, science came to the rescue in the meantime, and people developed a new method, right, to determine protein structure, which is cryo-electron microscopy. And for cryo-electron microscopy, you do not need a single crystal. All you need is the enzyme administration that you can then get diffraction pattern from single molecules of the enzyme on the surface using this cryo-electron microscopy technique. And with this technique, people have been able to get pictures of what the enzyme looks like, right? And this is what's shown here. And this is human soluble guanylate cyclase. This is the inactive form. So when NO is not bound, and you can see it's kind of a more globular structure. We have the sensor domain, the catalytic domain, and they're kind of folded together. And you can see when NO is bound, there's a huge change in the structure, right? In the NO activated form, the whole enzyme, it's like it straightens out, right? It's like when you're sitting at the table and you're slouching, and then your parents say, sit up straight. And then you sit up straight, right? And that's what this form looks like. So really interesting. So there's a huge conformational change between the inactive and the active form. And in this active form, the catalytic domain is turned on, and it will produce cyclic GMP. You can see this was published in 2019. So people started working on soluble guanylate cyclase back in 1980s, right? When NO signaling was discovered, 1980s. 40 years later, we have a cryomicroscopy image of the enzyme. We can see what it looks like. but no crystal structure using traditional methods could be obtained. But you can see now, aha, there is really something dramatic going on. So here's the reaction that the catalytic domain catalyzes. So when NO is bound to soluble guanylate cyclase and the catalytic domain is turned on, it will take up GTP and make cyclic GMP, right? And cyclic GMP is a secondary messenger in the human body used for all kinds of different things. And one thing that it does is that it induces relaxation of the smooth muscle tissue, right? By activating ion channels. And that's essentially now what we do. So the concentration of NO that is detected by soluble guanylate cyclase that is proportional to the amount of cyclic GMP that is produced by the catalytic domain, right? And ultimately that controls the level of dilation of our arteries. So that leads to vasodilation. And when you dilate the arteries, you lower the blood pressure, right? So now you can think about this if you have a patient with high blood pressure, right? And this is very common, especially it's genetic, but also older people suffer from high blood pressure. It's very dangerous. You can say, aha, nitric oxide to the rescue, right? Nitric oxide could actually be used as a drug, right, to lower blood pressure using this mechanism here. And that indeed is actually the case. And that actually goes back much longer than the research on nitric oxide, you know, in the context of human health. It goes back to the 1920s when people discovered that these kinds of molecules here, nitroglycerin, right, is an explosive. Nitroprusside is this inorganic salt of NO with 5 cyanides bound. And nitrite, these molecules, when you give them to a patient, they lower the blood pressure. And that was known since about the 1920s, 1930s. And so people started to use these molecules as drugs, as medication against high blood pressure, especially nitroglycerin, right, is the main part of TNT, an explosive. At the same time, you take it in to lower blood pressure. But people at that time did not know, why does it work? Nobody knows, right? Now we know. Now we know why it works. Because of that mechanism that I'm talking to you about, right? Using nitric oxide as a signaling molecule that activates soluble guanylate cyclase, produces cyclic GMP, and dilates your arteries. That's why it works. But for the longest time, from 1920 to 1980, 60 years, nobody knows why it works, but people take it because, you know, it improves human health. So this is a very interesting story here that's also related to that. Okay, so here is now, if we kind of zoom in more on the molecular level, right? How does it work? So in the inactive form, the heme in the sensor domain is ferrous, so iron 2, and it has axial histidine coordination. It's a 5-coordinate ferrous heme. And then when nitric oxide shows up in the environment, it gets coordinated to the heme, and it makes this 6-coordinate complex, right? Where we have 4 ligands in the plane from the heme, we have NO and we have histidine bound, so overall it's 6-coordinate. And this species here lives only a very short amount of time, and then we're cleaving off the histidine ligand here. The histidine comes off and the complex becomes 5-coordinate. And when the complex is 5-coordinate and the histidine comes off, that induces this large conformational change that we've seen in the cryo-EM images, right? And that turns on the catalytic domain and you start making cyclic GMP. So this is the mechanism how the activation works. And because the ferrous heme has a very high binding constant for nitric oxide, binding constant is in the range of about 10 to the 12 per mole, right? So this is a very high binding constant. And that's why we can actually detect nitric oxide at nanomolar concentration by using a ferrous heme, right? So this is how this works. And then after we detect it, by breaking the iron histidine bond, we can generate a signal, right? We can induce a conformational change, and we can use the signal then to turn on the catalytic domain. So, of course, we are chemists, right? And so we want to know everything, exactly how everything works. we want to know, how does this activation mechanism work? Why does the bond to the histidine break, right? If you do the same experiment with carbon monoxide, you can do that. You expose soluble guanylate cyclase inactive state to carbon monoxide, it will bind and it will form a 6-coordinate complex, but the complex will not break the iron histidine bond, it will not become 5-coordinate. So carbon monoxide can bind, but it cannot activate the catalytic domain. It cannot induce the necessary conformational change to turn on this enzyme, right? Only nitric oxide can do that, and that's because it can break the iron histidine bond. Why is it happening, and why does that work? And that is something that we also investigated in my laboratory. So this is kind of the research part I've been talking about that I can tell you about. Here's a stop flow experiment that further. So this experiment, shows this mechanism here. You can see this was published in 1999. So in the resting state here, the inactive form, we have the signal at 420 nanometers here. This is the resting state. Then when you add NO to that, this band disappears and a new signal arises at 431 nanometers. You can see this here. So after you add NO, 420 goes down, and you can see the 431 signal here coming up. And then this is this species here, right? Then we're breaking this bond and the complex becomes 5-coordinate, and then the band shifts to 399 nanometers, which is ultimately the signal. You can see then that starts rising here. So this stop flow data here indicate, the mechanism that I show here. So this is actually data that shows this process, binding of NO and then breaking of the iron imidazole bond. So this is the process I want to focus on now. This is the same thing again with a different cartoon, right? Again, here, this is the circle here. This is my heme, right? My heme cofactor. And we have this intermediate, a 6-coordinate, and then the sensor is activated when the bond to the histidine breaks and the heme becomes 5-coordinate. And you can see this is also a pretty old paper, 1992. It was postulated that this could be due to a so-called trans effect. Right? The trans effect in coordination chemistry means that the two ligands that are bound trans to each other, they influence each other, right? And so the bonding of the one ligand weakens the bond on the other side. And that was proposed, that this could be the electronic reason why the bond to the histidine here breaks in the presence of nitric oxide. And we wanted to study this. And so one way you could, of course, one way you can study this is if you could look at 5- and 6-coordinate complexes. So let's say you could prepare model complexes that are 5-coordinate and then the analogous 6-coordinate complex. You can compare their properties and you can study the nature of. This is very difficult to do in the enzyme because in the enzyme, this species here does not live very long. It's very transient, right? It lives less than a second and you make the 5-coordinate complex. But we can study this using model complexes where we can make the exact analogous 5- and 6-coordinate complexes and then look at their properties and see if we can learn something about this electronic effect. Right, so here are examples from model complexes, and these are the structures of 5-coordinate iron 2 NO complexes, and here are the corresponding 6-coordinate complexes, and these are parameters that were taken from small molecule model complexes. And you can see, if you just look at the iron NO unit, so this part here, you can see there's not a lot of a difference between them, right? These properties like iron N distance, 1.728 angstroms, 1.753 angstroms, these metric parameters are all very similar to each other. But one thing you notice is that in the 6-coordinate complexes here, the bond between the iron and the axial ligand is very, very long, right? And that, of course, in a way, is a reflection of this trans interaction here. That the ligand here in transposition to NO cannot bind very strongly. So the question is, how does that now relate to the properties of the iron-NO bond, right? And how does the iron-NO bond change between the 5 and the 6 coordinate complexes? So we can study this using spectroscopy. So here's one example. You can take an electron paramagnetic resonance spectrum, an EPR spectrum, because these complexes, have paramagnetic ground states. These complexes have S equals 1/2 ground states with one unpaired electron. And you can see the EPR spectra, they show some differences in their appearance. So we have this hyperfine coupling here from the NO ligand. But if you look at the G values, they are relatively similar, 2.1, 2.066, 2.01. And then in the 6 coordinate case, they're a little bit smaller. So there is some difference here in the amount of unpaired electron density on the iron, but otherwise they look relatively similar, right? So one of the interesting questions then is, what is the difference in electronic structure, right? If we go back here, what is the difference in electronic structure between these 6 and 5 coordinate complexes? And yeah, the EPR spectra do not show a lot there in that regard. They look quite similar, right? We also did MCD spectroscopy, again, looking at these complexes, these model complexes here. And in the MCD spectra, we see some differences. And in the 5 coordinate complex, we essentially see, a pure C-term spectrum, pure paramagnetic spectrum. But in the 6 coordinate case, we see these new signals coming up here. And these are essentially signals that indicate that the heme is becoming diamagnetic, right? And so the MCD data indicate that there is actually a difference between the 5 and 6 coordinate complex in terms of the distribution of the unpaired electron density. So that indicates that there is some difference here. But it's again, just looking at the spectra, it's hard to tell what it is. And so what ultimately really got us there was looking at vibrational spectroscopy, right? So vibrational spectroscopy is very powerful because the vibrational frequencies that you measure, for example, iron N stretching frequency, NO stretching frequency, these vibrational frequencies are proportional to the corresponding bond strengths, right? So if your, let's say, NO bond becomes stronger, then the NO stretching frequency goes up, like you compare two complexes. If the NO bond becomes weaker, NO stretching frequency goes down, right? So first of all, we can observe the vibrational properties, and we can again compare the 5 and the 6 coordinate complexes. We can ask the question, what's the difference between them in terms of electronic structure? Are there any difference in bond strengths, right? And that's something we can address using vibrational spectroscopy, and it's quantitative. We can also simulate the vibrational data, and we can calculate force constants for these bonds, right? We can calculate an iron N force constant, NO force constant, and then those you can directly compare between the complexes. So that's what we set off then to do. So if you're looking at examples from proteins, just to give you some relation here, for example, you can take ferrous myoglobin, right? You can bind NO to ferrous myoglobin, and you can observe the NO stretching frequency, just to give you an idea, about 16, 13 wave numbers. And if you do resonance Raman spectroscopy, you can observe one band at 547 wave numbers in the resonance Raman spectrum. Now, we'll go back here. That's a bit of a strange thing, right? Because you essentially expect to see free vibrations for the iron NO unit. You observe the NO stretch, of course, between the N and the O. You observe iron N stretch here, but there also should be a bending mode where the iron NO angle changes, right? So there should be free vibrations, not two. And so from the resonance Raman studies, we can only see two of these vibrations. And that's when we decided to use a new technique, which is called nuclear resonance vibrational spectroscopy. And I explain in a second what that is. But when we do nerves, we can finally see both features. Now we can see all three of them, right? We see the NO stretch, and then we see two features at lower wave numbers. But here the question is, of course, how do you assign them, right? Which one of them is the stretching mode, the iron NO stretch, and which one is the bending mode? And how do we tell the difference? And it's very important, right? So in myoglobin, this complex is 6-coordinate, and we can compare this to 5-coordinate complexes. And in the 5-coordinate complexes, the spectral assignments are easier. So in this case, the iron NO stretch is about 530 wave numbers, right? So now this becomes very important, right? So when we go from 6-coordinate to 5-coordinate, if the 547 is the iron NO stretch, you can see that it's about the same. So the iron NO bond does not change much between the 5 and the 6-coordinate complex. But if this is the iron NO stretch at 443, then you can see when you go from the 5-coordinate to the 6-coordinate complex, the iron NO bond becomes much weaker. So assigning these two features correctly is very important in order to understand the changes in the electronic structure. And again, we went on then to use, as I said, the model complex to study this, because with model complexes, we can study the exact analogous 5 and 6-coordinate complexes. So we can use this ligand here. This is called the tetraphenyl porphyrin. This is a synthetic porphyrin, not what nature uses, right, but synthetic. And then you can make this as a 5-coordinate complex without the imidazole, or you can make this as a 6-coordinate complex with the imidazole bound, and then you can compare the properties. And as I said, in the 5-coordinate case, the iron NO stretch is about 530 wave numbers. And so now really the question is, what about this 6-coordinate complex that's shown here, right? What are the vibrational data for this kind of 6-coordinate species? So we're going to use now this new technique, which I call NERVS. So I want to talk about what that is first. So NERVS is nuclear resonance vibrational spectroscopy. And this is a technique that we can only use at a synchrotron, at a beamline. So here's one example. This is a synchrotron that is very close to the University of Michigan. This is Argonne National Lab. And this is called the Advanced Photon Source. And at the Advanced Photon Source, they have a very bright beam. And so they can generate X-rays, you know, at very high intensity. And in nuclear resonance vibrational spectroscopy now, we're looking at the nuclear transition of iron 57. So we have to prepare our complex with iron 57, the 57 isotope of iron. And that, of course, is the Mössbauer active isotope, right? Mössbauer spectroscopy measures the nuclear transition between the nuclear ground state and nuclear excited state that is metastable of iron 57. And that's traditional Mössbauer spectroscopy. Here what we do is we actually measure inelastic scattering. So we measure the inelastic scattering that comes from exciting the complex at the Mössbauer line. And in order to do that, we need a very bright X-ray beam, and we need the exact light energy, which is 14.413 kiloelectron volt or energy. That's the Mössbauer line. And so we need this X-rays with this energy, and we need to excite the nucleus of iron 57. But now we measure inelastic scattering. So in a way, then, this is like a nuclear resonance Raman spectroscopy, right? Not normal Raman spectroscopy where you have electronics or electrons scatter. The visible light here, we actually use nuclear scattering. So we measure inelastic scattering in resonance with the Mössbauer line of the iron 57 nucleus. And that gives us a vibrational spectrum. And the special thing about this technique is that this technique has no optical selection rule. And so that means that all the vibrations where iron is moving, all of them can be observed with this technique. And the intensity is proportional to the amount of iron motion, or the square of the amount of iron motion in your normal mode, right? And so that means all the vibrations where the iron is moving can be detected, and there is no other selection rule like in IR or Raman. Like IR, you have to have a change of the dipole moment, or in normal Raman spectroscopy, you have to have a change of polarizability. There's no such selection rule here, right? So it's really the only selection rule in that sense is that the ion has to move. So this technique is very good for detecting metal ligand vibrations and ion complexes. You can see here at the beamline, this is a very complicated setup where we have the beamline itself produces the X-rays, but they have a high intensity, but they also are not monochromatic. And then they have built at Argonne National Lab, at the APS, they built this monochromator system that then allows you to get down to a resolution of about 1 millielectron volt. And that's about 8 wave numbers. You can see it's a little bit better than this. So our resolution is about 6 wave numbers with this experiment. And this is what the data look like. So here's example for nerves data now for this model complex, right? This is this guy here. So the iron tetraphenylporphyrin complex with 1 methylimidazole bound. Gives these kind of data here. And then we can prepare the complex with normal nitric oxide with isotopically labeled nitric oxide. And when you label with isotopes, so you make the mass higher, that means the vibrational frequency shifts lower, right? And so we can label the NO, we can identify all the vibrations that have contributions from the NO ligand to it. And you can see there are 2 modes that are isotope sensitive. One is at 563 wave numbers and one is at 437. Right? So that confirms the data I mentioned before. But we still don't know, we still don't know how to assign these vibrations, right? We can see that, yeah, we can identify these 2. One is the stretching mode, one is the bending mode, but we still don't know which one is which. But now we can do a further trick, and that is we do an experiment with single crystals. And we did that in collaboration with Bob Scheidt's group at the University of Notre Dame, right? Where you make a single crystal of your complex, and then you orient the single crystal in the beam, and the beam is actually polarized. So you can do experiments like this. So we have linear polarized light, coming from our light source, from the synchrotron, and we can take the crystal and we can rotate it in a way that the plane of the heme, lines up with the direction of oscillation here of the electric field vector, right? And when we do this, then only vibrations get excited or scatter or contribute to the spectrum where the ion moves in the plane of the heme. And that gives that spectrum here, is shown here in blue. And then we can rotate the crystal and we can measure again. And now we rotate the crystal. So we're perpendicular, to the direction here of polarization of the incoming beam. And now only vibrations can contribute where the ion moves out of the plane of the heme. Right? And so by doing the single crystal experiment, we can distinguish vibrations where the ion moves in the plane of the heme or where the ion moves outside of the plane of the heme. And of course, for the ion NO stretch, the ion will move perpendicular to the plane of the heme. For the bending mode, it will move in the plane of the heme. And so if you look at the data, you can see the band at 437 wave numbers has strong out-of-plane intensity and almost no in-plane intensity. So we can see this is the ion NO stretching mode, where the ion moves out of the plane of the heme. Okay? And 563 is the other feature, right? The other feature that is isotope sensitive. And you can see here we have a mixed polarization. So this vibration has contributions from in-plane and out-of-plane motion. And what that means is that essentially the ion, if this is the plane of the heme, the ion doesn't move like this or like this. It kind of moves diagonal. So the ion moves a little bit out of the plane of the heme And that means we have some intensity in this out-of-plane polarized spectrum, some intensity in the in-plane polarized spectrum. Okay. So that's pretty good. But in order to get this further, we have to also simulate the data. So we can use our quantum chemistry-centered normal coordinate analysis. Normal coordinate analysis is the standard method of simulating vibrational data using a force field. And because there's a very simple relationship in nerves between, intensity and the properties of your normal mode, we can actually simulate spectral intensities here as well. So we worked very hard, and you can see here we were able to fit the data. So the spectrum in black is the experimental spectrum, and the one in orange is the simulation. And then in the simulation, we can also look at the polarizations, and you can see that we can reproduce in the simulation the fact that the 437 mode is strongly out-of-plane polarized, and the 563 has this kind of mixed polarization, right? So we're able to reproduce these key features of the spectrum in our simulation. So that means we can now make our assignment, right? We assign the 437 wave number feature to the ion NO stretch, and this one to the bending mode. And we can also animate that. So you can see here, you can see this a little bit better, right? So this is the stretching mode. You can see the ion essentially moves perpendicular to the plane of the heme. And you can see this is the bending mode. And you can see in the bending mode, indeed, the ion moves kind of in between, right? And then in the bending mode, most of the oscillation is actually in the nitrogen. And when this is the mode at 563. And these are the pictures that we will obtain from our simulation of the data. Okay, so we can skip this one here. We can bring it all back together into this slide here. This is the comparison now for this particular model complex, right? So this is the 5-coordinate complex, and here are the vibrational data with the NO stretch at 1697, the ion NO stretch at 532, and the bending mode at 371. And these are the force constants that you calculate from the simulation. And here's the 6-coordinate complex. So we had to work very, very hard, as I showed you, and use nuclear resonance vibrational spectroscopy. And we even had to do single crystals, just so we can make this assignment here. Right? The ion NO stretch at 437, the bending mode at 563. And what you see here is that in both cases, the NO bond and the ion NO bond become weaker in the 6-coordinate complex. So when you inverse the process in a way, you start with the 5-coordinate complex and you bind imidazole in transposition to NO, both the ion NO bond and the NO bond become weaker. And you can see this in the vibrational frequencies, and you can see that also in the force constants, right? And so we call this a direct correlation of the NO and ion NO bond strengths. And this direct correlation is a hallmark of bonding. I'll show this to you in a second, right? So what that means is then that the electronic effect that we're looking at for the activation of soluble guanylate cyclase is a fact that comes from the ion NO sigma bond. And that gives rise to this direct correlation that we observe in the vibrational data. So we can deduce that. So we can look directly at the electronic effect that's responsible, for this trans effect. So then we can take a look at the principal bonding scheme between an ion porphyrin and NO. And this is very, very crude, but it kind of shows what's going on. So in these kinds of complexes, ion is in the ion 2 state, it's in the low spin state, which means that the T2G type orbitals are here fully occupied, the other ones are empty. And then here's our NO, and NO, of course, is a radical. It has one unpaired electron. And when the NO approaches the ion center, you will start forming the sigma bond between the DZ square orbital of iron and this pi star orbital of the NO ligand, right? And that is then here indicated in this orbital here. And it turns out this is the key orbital, right? This is the singly occupied molecular orbital of the complex that carries this one unpaired electron that the NO brings into the complex. So this constitutes the sigma bond. And then on top of this, we have this empty pi star orbital here, which can make a pi back bond with the iron center. And in my coordinate system, it's the dyz orbital. So here would be the bonding combination of this orbital, called this pi star B for vertical, and this pi star H for horizontal. This is the bonding. This is the anti-bonding orbital for this pi back bond. Okay. But based on the vibrational data here, we know that the change in bonding comes from the sigma bond. So the sigma bond here changes between the 5 and the 6 coordinate complex, and that is the electronic origin of this trans effect, right? That I mentioned. So if we, this of course is just simple, you know, MO diagram drawing. If we want to make a closer comparison now, we have to look at the actual orbital, right? So now we can use quantum chemical calculations. We can calculate what this orbital here looks like. And here's someone talking. Maybe the person could mute the conversation. Okay, so we want to see now how the sigma bonding changes between the 5 and the 6 coordinate complex. We should look at quantum chemical calculation, DFT calculation, right? And so this is what this slide shows. This is the 5 coordinate complex, and you can see, you know, this is the same as this orbital here. This is just a little chem draw. This is the real thing now, right, from the calculation. So you can see that we have a very strong bond. This is the pi star orbital here of NO. Here's NO. Here's iron. This is the DZ square orbital here, and we get a very strong bond. And that unpaired electron, right, this orbital is occupied with one electron here. That electron is fully delocalized over the iron NO unit. So this orbital is essentially 50% iron, 50% NO. And these are the spin densities. So spin is about 50% on iron, 50% on NO. Now in the 6 coordinate complex, you can see that there indeed is a pretty dramatic change in the property of this orbital, right? So the first thing you notice is that the interaction with the DZ square orbital becomes much smaller, right? You can see this orbital is about 50-50, but here the contribution from DZ square is smaller. And you can see it's also kind of, you know, instead of standing up straight, it's kind of tilted away a little bit from the NO ligand. So this sigma bond here becomes a lot weaker in the 6 coordinate complex, right? And on top of this, you can see that in this orbital here, the interaction between DZ square, and this is the sigma donor orbital of our imidazole ligand here, which we use instead of histidine in the model complex. You can see this interaction here is actually anti-bonding, right? So this orbital here, of course, is empty without NO, and when NO binds, this orbital becomes singly occupied, and it is anti-bonding with respect to the iron and the imidazole bond. And that is a direct reflection now of this trans effect, right? So the binding of NO weakens the iron imidazole bond because we're occupying an orbital that is iron imidazole anti-bonding in nature. That means the iron imidazole bond now will become extremely weak, and in the enzyme, in soluble guanylate cyclase, it will break, and that induces the conformational change that activates the sensor, then the catalytic domain, to make cyclic GMP. So we can use these exact comparison between these model complexes to understand the electronic effect that leads to the activation of soluble guanylate cyclase by NO binding through this trans effect, right? You can also think about the trans effect as a competition, right? So we have the DZ square orbital here, and we have two ligands, NO and our imidazole, and they compete for binding to DZ square, and NO makes the stronger bond, so it ends up in the bonding combination, but the imidazole makes the weaker bond, it ends up in the antibonding combination, and that weakens that bond. So if we go back then to slides, I guess I should have, yeah, here it is, right? So the trans effect here that breaks this bond, we can call this a sigma trans effect, and it's mediated by donation of NO into the DZ square orbital of the heme, which weakens the ion imidazole bond, it breaks, and then that leads to the formation of the 5-coordinate complex, and that activates soluble guanylate cyclase. Okay, so that's one interesting aspect of this. Another interesting aspect is if we make this comparison with myoglobin. I don't want to dwell on this too long, but we're also almost out of time. So we can compare the model complex to myoglobin and these different active site variants of myoglobin. And what you find is that the model complex is very similar to myoglobin mutants where the distal histidine has been removed. You know that in myoglobin, we have a hydrogen bond, right, to the diatomic bound to the ion center. And if you remove that histidine responsible for this hydrogen bond, you essentially have something very similar to the model complex, which of course does not have this hydrogen bonding either. And that's what's indicated here in this vibrational correlation plot, where we're looking at the NO stretching frequency versus the energy of this bending mode. And we can see that the model complex lies in this quadrant here, where we have these myoglobin mutants. This is another interesting aspect of this as well. So the bonding is influenced by hydrogen bonding in myoglobin. Now, that's not directly relevant for soluble guanylate cyclase, but it is another thing that you observe when you kind of look at these vibrational frequencies. In any case, I better wrap up now, otherwise we don't have time for questions. All right, so here's my summary. So I showed you that we were able to get vibrational assignments for the 6-coordinate ferrocene nitrosyls using this technique called NERVs, and we're using single crystals to do this, and we can assign our spectra. And the most important thing is that we can now analyze the trans interaction between NO, right? Here, oops, my mouse is jumping, between NO and the trans ligand, the imidazole, right? And we can understand the nature of the trans effect, so that binding of NO here to the heme induces this antibonding interaction between the ion and the imidazole. And you can see with the arrow here, right, that induces breaking of this bond, which moves this alpha helix here further away from the heme. And that essentially, this motion then induces the conformational change in soluble guanylate cyclase that leads to the activation of the catalytic domain. So this whole helix here swings back when the histidine falls off the ion center here. So the binding of NO through this sigma trans effect causes the breaking of the ion imidazole bond, and then that activates soluble guanylate cyclase. So one of the other aspects then is, because imidazole binds so weakly, right, to iron, it is actually challenging to make these 6-coordinate complexes in the model system, and you have to add a large excess, usually of the axial ligand, in order to form these kinds of complexes, because their binding is so weak because of this sigma trans effect. But you can help yourself out then by making constructs like this, and this is something that we did in my lab. We made this complex where we attached our imidazole group to this tail. And now this is a little bit like the enzyme. In the enzyme, you have an alpha helix here, right, where the imidazole is connected to, or the histidine, and that holds it in place. So it keeps it bound to the ion. And here we use this kind of covalent linkage, and we can kind of keep the histidine bound to the ion here. But I don't want to talk about this too much, because we're essentially, I think, out of time. So then let's go to my final slide. my conclusion slide. So here's a recent picture of my research lab. And I want to, of course, thank my students for all their hard work, right, over the years. These studies, if you looked at the publications, this all goes back to around 2006 to 2010 is when we really worked on this to again understand the nature of this trans effect or trans interaction that is at the heart of activating soluble guanylate cyclase. And we have since then moved on to do other things. And want to thank all of you for your attention and thank you for listening to my presentation. And I hope that this combination of, you know, some basic bioinorganic chemistry and then a research component at the end, I hope that that was entertaining, and I hope that you could learn something interesting about nitric oxide. So thank you very much for listening to my presentation.
Yes, thank you, Prof. Lehnert, for the nice presentation. Yeah.
Thank you.
Now I will open the question and discussion session. I found some question in the chat box, and I will try to present here in the slide.
Okay.
Maybe. The first question I will read here from Mr. Isa Ansori from ITB, Institut Teknologi Bandung. Yeah. He is from physical engineering. Yeah.
Okay.
Biomedical engineering.
Oh, biomedic engineering. Yeah. Sorry, biomedic engineering. He's still on here. Sorry, that is a bit hard for me to understand the chemical mechanism. And I would like to ask your suggestion about what we need to prepare when we want to develop the NO sensor, for example, based on electrochemical, plasmonic, or KGM. Thank you.
So I think that having a heme, right, the heme group is a very good sensor group for nitric oxide, and that's why nature uses it, because it has a very high affinity for NO, as I mentioned, with binding constants that are going up to like 10 to the 12 per mole or something like this. And so if you want to build a sensor, you could think about how you can incorporate hemes into your essentially the electrode surface, right, of your sensor. So it could be an electrochemical sensor where when NO binds, it will change the redox potential of the heme, right? So that will be one way. Or of course, you could use a microbalance, although this is probably difficult to do in the field. So electrochemical is probably better. You have the hemes that are 5-coordinate, and when NO binds, it will change the electrochemical potential. Now, one of the problems is, of course, that iron 2 is also oxygen sensitive, right? And so the problem then is, if you build a sensor from iron 2, you cannot use it in air because iron 2 is oxidized by dioxygen to iron 3, right? And then it will not bind NO so well anymore. But here you could do something that nature doesn't do. And you could use a cobalt porphyrin. So cobalt porphyrin also binds NO very strongly, but cobalt 2 is not oxygen sensitive. So if you use cobalt porphyrin instead of iron porphyrin, you could build an electrochemical sensor for NO that would not be sensitive to oxygen. I don't know if it would be sensitive to CO, but it would not be sensitive to oxygen. And so then you could have an electrode, and you would have to make a film on the surface of the electrode made of some kind of cobalt porphyrin material. And then you could use the electrochemical potential that change when NO binds to detect nitric oxide. And so in that way, you could kind of employ the principle that nature uses, but you would generate a different signal. So instead of generating a conformational change, now you generate a change in electrochemical potential, and you can measure that at very high accuracy, with cyclic voltammetry or some other measurement. You could measure electrochemical potentials for very high accuracy.
Okay, thank you very much for the answer. I will
Is
try
there
to
any
review
feedback
this one.
from Dr. Isa? Is there
Yeah,
any
no,
feedback?
no. Actually, I would like to first searching about the dimensions techniques from Prof. Lehnert, and maybe later, yeah, if I have some curiosity, I would like to send email probably.
Yes, yes,
Later for
please
one.
do that.
Yeah. Thank you.
Yeah, as Vitri mentioned in the introduction, we do a lot of spectroscopy. So we use EPR and MCD, and we use this beamline technique that I was talking about, nerves, very much. So I know that might be a little bit of a challenge for students, but I was trying to emphasize, what do we learn from these techniques, and try to emphasize that more, because the techniques are very complicated. But yeah, please feel free to send me email. I'll be happy to answer.
Okay, nice. New contact here, yeah. New contact one, Dr. Isa, to improve our research.
Yeah, that's why.
Okay, thank you. Yeah. And maybe we can move to the next question from Prof. Sayakti. Yeah. Dear Professor Lehnert, how does it happen, or is it predicted when an activation of SGC and ligand is replaced with the carbonyl, with the CO, yeah, or thiocarbonyl, GS, or isonitrile, NCR? And the next question, what is the source of an ligand in the body?
So the source of NO, as I mentioned in the first part of my presentation, is L-arginine, right? So L-arginine, amino acid, is metabolized by nitric oxide synthase, and that is the way that nitric oxide is produced in the human body. It was very surprising back in the early 1980s when this was discovered, right? Because nitric oxide is so toxic, nobody would have thought that our body would make such a toxic molecule, right? Inside our body, such a toxic molecule is produced. But that's what they found, right? And that's what delivered the Nobel Prize for them. So nitric oxide synthase, as I described in the first part, oxidizes, hydroxylates, oxidizes L-arginine to citrulline, and then releases nitric oxide in that process. That's how it is produced in the endothelial cells. If we go back, I'm just going to do it by hand. Leave the spectroscopy behind, my favorite thing, and go back to the slides. Oh yeah, here's a good maybe one, right? So here in the endothelial cell, there is NOS, right? Oxidizing L-arginine to citrulline, nitric oxide is generated. It is released in this process, and then here it is detected by soluble guanylate cyclase, and then that makes cyclic GMP, and then that essentially induces the relaxation of the smooth muscle tissue. So it's kind of a signaling cascade that starts with NO being detected here in SGC.
Okay. Is there any feedback from Professor Sayakti about the question and the answer?
Yeah, and the first question, please.
Oh, the first question, yes. The first question is about how does it happen, or how is predicted when an activation of SGC and ligand is replaced by with the carbonyl, yeah, thiocarbonyl or isonitrile.
So
NCS, yes.
yeah, so if you go back to, yeah, this may be a good one. If you replace with CO, carbon monoxide will bind to the heme, or isocyanide will bind to the heme. But in both cases, the 6-coordinate complex is stable. And people have, I don't know if people have bound isocyanide, but people have bound CO to soluble guanylate cyclase. There's no activation because it stops here, right? With CO, you get stable 6-coordinate complex, it stops. It does not break the iron imidazole bond or iron histidine bond, and it does not activate SGC. So you just bind CO and nothing else happens. And isocyanide would be the same, but I don't know if people have actually tried it on the enzyme. But both of those ligands do not have this strong sigma trans effect that NO has, and so they cannot activate soluble guanylate cyclase.
especially if replaced with carbonyl.
Yeah, with carbonyl, there's no activation because this 6-coordinate complex is stable with carbonyl.
Yeah.
No further reaction happens, and so that means there's no activation. You have to break the bond here. You have to break the bond to the histidine in order to get the activation, and only NO can do that because of its special electronic effect.
Yes. Because different activation for sigma or p bond.
Yeah, so CO is weak sigma donor, right? And
weak
so
sigma donor,
yeah,
weak
so
sigma
it cannot,
donor and could be acceptor, but in O it's not like that.
NO is a strong sigma donor.
Okay.
Yeah, and you need a strong sigma donor for the activation.
That is a key. That is a key for this.
That is the key.
Okay.
Yeah.
Okay,
and
thank
so
you.
That's
thank you.
Yes. Thank you so much.
Okay.
Yeah.
Yes, nice. Thank you, Prof. Cyclase and Prof. Lehnert for nice discussion here. And I will continue for the next question from Pinka. Dear Professor Lehnert, I would like to ask about the NO application. Can nitric oxide be used to treat the several COVID patient for a long period of time, maybe related to some cases that have been reported that COVID also affect the brain in some people? Is the application of NO in the COVID patient is possible also to treat with this NO, Prof. Lehnert?
Yeah, that is true, but that's a different function, right? So NO is used as a drug to lower blood pressure, as I mentioned, but you don't give the patient NO directly. You use NO donors. Nowadays, we have more sophisticated ones, but back in the day, as I mentioned, nitroglycerin is used. Now, for biomedical application, a big focus is the fact that NO is antimicrobial, right? So as I mentioned in the beginning of my presentation, nitric oxide is produced by our immune system to kill pathogens, to kill bacteria that infect our body, right? And so now we can use nitric oxide in the clinic. And so, for example, nitric oxide is used in inhalation therapy, right? So patients who suffer from pneumonia, so infection of the lung, they can breathe in low doses of NO in order to try to kill the infection, making use of the fact that nitric oxide is toxic to bacteria, right? And in fact, the inhalation therapy has also been tested for COVID, and there were some beneficial effects. I don't know if it's used widely to treat COVID, but I read a couple studies that said that, inhalation therapy can help to clear COVID from the lungs, and it can also help with the problems. One of the problems of COVID is that you get blood clot formation, in your small blood vessels in your lungs. And nitric oxide, because it inhibits, blood clotting, it can also help with that. So there are some beneficial effects from inhaling nitric oxide at very low doses, of course. Otherwise, it kills you, right? So you have to use a low dose. It can actually help with infection, and it can help with COVID. But I don't know if it's widely used in the clinic for COVID patients. That's a very good question. Does that answer your question?
Pinka, maybe do you have feedback, Pinka?
Oh, okay, I understand. Thank you, Prof.
Oh, nice. Smart student. She understand already, bro.
Good. Need student questions too.
Yes, it's nice. And the next question from Septia. She is the master student from our department. Septia from UNS. I would like to ask about the concentration of NO inside our body. It is stated that before the concentration is very small inside our body, but what if the concentration is too low or too much? And what's the effect to our body? Thank you.
So the concentration of NO is very low. It's like nanomolar, sub-nanomolar concentration. And that is the the normal concentration of NO when our body is healthy. Now, if you do not have enough NO, so if, let's say, for some reason the nitric oxide synthase doesn't work properly, or there's something else, if you don't have enough NO, then you get high blood pressure, right? That's exactly the problem, because now there's not enough dilation of your arteries. The arteries will get smaller, and that means, for one thing, you'll get high blood pressure, and you get memory problems. Memory loss is one of the other things, and that relates back to neuronal NOS, right? So the nitric oxide synthase in neurons in the brain is involved in signal transduction in the brain. So too low NO is not good for the body. Too high NO, though, is even worse, right? Because nitric oxide, at the end of the day, is very toxic, and nitric oxide becomes toxic at about micromolar concentrations. And so if you suffer, for example, sepsis, right, is a famous example. Sepsis is infection of the blood. So you have an accident, you know, you play soccer, you have an accident, and you break your leg, and you have an open wound, and so bacteria get in, they infect your blood. Now your body will respond with the immune system. The immune system, of course, uses NO as an antimicrobial, but now, because the infection is in your blood, there will be a lot of NO produced by your immune system, too much, right? And then that can lead to organ failure and organ degradation, you know, kidney, liver damage, and ultimately can be very dangerous. So too much NO is very dangerous. And over time also, in, you know, if you have a chronic infection, so let's say you have a chronic infection in your knee, then your immune system will keep producing about micromolar concentrations of NO to kill this infection, but it cannot. So it goes on for a very long time. It will damage the cartilage in your knee. It will damage the bone from these longer periods of higher NO concentrations, and then it can lead again to other bad effects, right? And so, yeah, too much NO is very dangerous, and there are a couple of diseases that are related to that as well. So it's important that the NO concentration is kept under good control in our body.
Yes, it's very well explanation here. Septia, do you have feedback about this question?
Yes, Mrs. Witri. So how to adjust the concentrations of the NO in our body, or we just living healthy life, and then the concentration will be at its exact amount inside our body? How to adjust so it will be not too low or too high or too low concentrations? Thank you.
So there are some drugs that have been developed by the pharmaceutical industry for problems with too high NO levels, and these are drugs that essentially target nitric oxide synthase, and these drugs they lower the activity of nitric oxide synthase. So there is some medicine available if you suffer from that problem. It's a particular problem in diabetes. So patients who have diabetes, you know, they oftentimes suffer from problems with also NO, and too high levels of NO that's actually produced by neuronal NOS, but not in the brain. It's produced in your nerve ends, in your legs and your feet, and that can lead to chronic nerve damage. And so there are some drugs that target NOS and lower its activity, and they're able to lower the amount of NO that is produced. So there is some ways to do that, because there are certain disease states where you have problems with nitric oxide. However, if you don't have diabetes and you're healthy, then you eat a good healthy diet, I think you'll be fine. So then you don't have to worry about it. But there are some diseases that lead to problems with NO concentration. Yes, that is correct.
Okay.
Okay, sir. Thank you for the answer of my questions. Thank you so much.
Yeah, you're
Okay,
welcome.
thank you for the nice discussion. And the next question, yeah, Prof. Lehnert from Dr. Teguh Indah. I have an interest on NVRS, yeah, nuclear resonance vibrational spectroscopy. What is actually the difference with the conventional Raman spectroscopy, and what can we obtain using this instrument compared with the conventional one? Is there any requirement for sample? Yeah, only purpose for specified sample or something like that?
So yeah, conventional Raman spectroscopy, of course, uses light scattering from electrons, right? So you excite with visible light or near infrared or UV light, and electrons are scattered. The light is scattered by the electrons in your molecule. So this is a technique you can apply to any molecule. Nerves is scattering by the nucleus, and it's nuclear scattering, and it requires you now to excite a nuclear transition. So it's essentially something like nuclear resonance Raman spectroscopy, if you want to look at it like that, right? In resonance Raman spectroscopy, you excite electronic transitions with your laser and you get the resonance scattering. And in nerves, you now excite the nucleus. And so you need, first of all, a source for X-ray or gamma radiation. So nerves cannot be done in a laboratory. You have to go to synchrotron to use that technique. And then it only works for elements that are Mössbauer active, right? And so, for example, for iron, it only works for iron 57 because only iron 57 has a metastable excited state of the nucleus. So then that means you have to prepare your complex with iron 57, not just normal iron, which is much more expensive. So that is a problem. And it doesn't work for cobalt or manganese or other metals, right? So you cannot do it. With normal Raman spectroscopy, you can apply to any complex, but nerves, you can only apply to Mössbauer active nuclei. So iron 57, you can do tin, you can do gold. So there's only some select elements that you can do, and you have to do it at the beamline, right? And there are only certain synchrotrons that actually are bright enough to do nerves, and there's only, I think, 4 or 5 in the world where you can actually do this. And so this is a problem for access everywhere. We are lucky because in the US, there is one place in the US where you can do nerves, which is at Argonne National Laboratory. That is in the Chicago area, and that is about 4 hours away from where I live, at University of Michigan. So we have a good access there. There is one place in Japan where you can do it, Spring 8. There's one place in Europe where you can do it. And there's a new beamline in China, forgot the name, where you can do it. So there's only 4 or something locations in the world where you can use this technique. So access is a big problem. But then you can see from my data, you can measure, you know, all vibrations that contain ion motion. So all metal ligand vibrations you can measure very easily. And then, you know, using labeling or single crystals, you can then analyze your data very nicely. You can simulate it very nicely. But yes, getting the data is not easy.
Yes, that's the next problem. And what is the requirement for the sample? Is only applicable for a single crystal?
No, you can do powder sample as well. It doesn't have to be single crystal. You don't need a lot of material. So that's nice. You need like maybe 10 milligrams, 15 milligrams of material. Mm-hmm. So that's not a problem. But the problem is you have to prepare it with iron 57, right? Not natural abundance iron. Iron 57 is very expensive. Yes. And so that is another barrier, right? In order to use that technique.
Okay. And Dr. Teguh Indah is here. Is there any feedback, Dr. Teguh?
No, I got the answer already, Dr. Witri. Thank you very much, Professor Lehnert, for the answer.
Thank you.
Okay. And the next question here From Larasati. Once I have ever read about the role of nitric oxide in cancer. Does it act the same way? It to control the blood pressure? It prevents cancer grow, but in some cases it also promotes the growth. This is this duality. What's mechanism is most likely happen actually?
Yeah, that's a very good question. I think a lot of people would like to know that. So yes, nitric oxide has a whole bunch of other interesting functions in the human body, and it's concentration and location dependent, and it's not always clear. So yeah, so nitric oxide can induce apoptosis in cancer cells. So there are some people that design drugs against cancer, you know, that these are molecules that carry NO, and then maybe you can shine light on them or something, you can release NO, it can induce apoptosis and kill cancer cells. And those will be at higher concentrations, but at lower concentrations, it is not clear how it affects the cancer. And so there are also some studies, as you're saying, where nitric oxide actually promotes the proliferation, right, of cancer cells. And so it might also depend on the type of cancer. I'm not an expert in that area, but yes. So people are really interested in to see if nitric oxide-based drugs could be used for cancer treatment. There's a lot of experimental stuff going on right now, you know, all over the world, using dinitrosyl ion complexes, you know, giving them as drugs. There's a lot of studies on animals right now. But the effects overall look promising. So most of the stuff I've seen shows that nitric oxide can in fact kill cancer cells. And then the question is, can you make a drug that could be given to a human that will kill the cancer cell, but not the human? And I think that is still a big challenge in that field. A lot of these compounds that people use to deliver nitric oxide are also toxic.
Larasati, do you have any feedback?
No, thanks. Mrs. Fitri.
Oh, okay.
I think it is still debatable because in the same time it promotes growth, but it is only little studies, I think. But most studies what to say? They state that it prevents the cell proliferation.
Yeah, I mean that's a lot of the stuff I've seen, is that it prevents cell growth, it induces apoptosis, and it slows down proliferation. So there's more indication of anti-cancer effects for NO, but it might also be concentration dependent, right? And so you need maybe a higher concentration to kill a cancer cell, and a lower concentration maybe it can actually promote it. So I don't have a good answer yet. It's a difficult question. I think a lot of people would like to find out. There's a lot of research going on in that area.
Okay, thank you, Prof.
Okay, I think the question here, I have read all the questions. Yeah. Please, the audience, if you want to ask directly by open your mic here, you can raise your hand, and then I will offer for one more question here because the time are limited also. In the US, what time is it now, Prof. Lehnert now?
It is 11:15 at night.
Oh, 11:15, yeah.
Getting late.
Getting late, yeah. I think the lecture is quite long now.
May I have
no?
one final question?
Yes, final question, Prof. Fitri. Prof. Fitri.
All right.
Can you please inform the opportunity to get scholarship from your university, or maybe the availability for Master of PhD student in your lab?
So in the US, if you come for a PhD, then you don't have to pay for that, right? So if you join as a PhD student, then you will actually get paid a salary, and the PI or your PI pays your tuition. So for PhD, there is no cost for the student, but you actually get paid a salary to make a living. So that is that's very good. However, for master's degree, it's very expensive because for master degree, you have to pay, but for PhD degree, your boss pays you, right? And so always that's the better option for everyone, not just international student, but also domestic students. So yeah, so there's always research opportunities in my lab. I have a lot of students, a lot of stuff going on. In the US, you can only apply to the PhD program once a year. It's very different from Europe, right? In Europe, you apply to an individual professor, individual PI, and then they can pick you up and they can hire you and you go to their lab. But in the US, it doesn't work like this. In the US, only once a year, the department recruits students as a whole. So there's no recruiting by individual professors. You have to apply to the PhD program in the Department of Chemistry at the University of Michigan in general. And that can only be done once a year, which is deadline is around December. And then when you get admitted, you come to Michigan and then you find the research group. So it's a very different system from the way it works in Europe. So you have to keep that in mind. But yeah, I mean, the deadline is in December, so it's October now. So you still have 2 months to write an application. For international student, the TOEFL is required, right? You have to provide the TOEFL score when you apply as an international student. So that could be a little bit of a problem because you can only, I don't know how often you can take the test. So it's maybe the time is too short between now and December to get a TOEFL score. That's really the only requirement. And then of course, yeah, you have to have a bachelor or master degree in chemistry in order to apply for a PhD program, right? Minimum requirement is bachelor degree. And it's good if you have some research experience as well, because the department likes to hire a student who has some practical research experience in the laboratory as a preparation for the PhD.
Okay, thank you, Professor Lehnert, for your information. I hope students have noted that information very well. Thank you.
Yes, thanks, Prof. Fitria. Maybe if you also have interest to apply the Fulbright Visiting Scholar Program, now it's open and the deadline is until November. You can stay in the US around 3 until 6 months, I think, if you want. Yeah, I have the link that I shared in the chat because it's from the International Office of our university. If you want to apply also for the lecturer.
Oh, okay, that's good. So you actually have like a fellowship, so students can come to the US for research, like internship. Yeah, that's very good.
Yes. Oh, nice lecture today. We have discussed a lot of things from the side of chemistry and then spectroscopy application in the biomedic and also in the biomedical technique like for sensor, for example. And hopefully we can have the lecture again maybe for the next semester or something like that. And I thank you, Prof. Lehnert, for your kindness to give a lecture to our department and also for the audience for the time. And please can you close the slide, and I will share the certificate of appreciation. Yeah.
Oh.
Wait, I will share here. Here is we would like to appreciate to give the certificate of appreciation to you, Prof. Lehnert. Here is our Vice Dean of Academic Affairs, Prof. Suryanti, and also Prof. Fitria. And our program today is actually a part of WCU program, World Class University program, that hopefully will increase our internationalization in our university. And in the future, hopefully we will create some new network to conduct academic activities like for teaching or researching. Yeah.
Yeah, that will be very nice. And yeah, thank you for the certificate. Looks very pretty.
Okay, I will send this file by email. Yeah.
Yes, please.
And
yeah, I can next semester I can teach again. I can refine my lecture. You can tell me which part you like better, and I can refine my lecture for next semester.
Okay. Start the sharing. And before we close session, maybe we will take the photo session for all the audience and the speaker. Please open your camera, the audience, as documentation. Yeah.
Okay.
Please open the camera for the audience. There's Mr. Agung also. Mr. Agung is what is our senior lecturer from mechanical department. And he stay in Japan very long, Mr. Agung. How long have you been in Japan?
Yeah, thank you very much, Prof. Lehnert, for your interesting lecture. Yeah, I have to study more actually.
Thank you.
Yeah, we take the picture. Oh, there's still a black side here. 1, 2, and 3. Some audience are already living here. Maybe because they have also another activity. And the second slide, 1, 2, 3. Yes. Once again, thanks for the audience, also for joining this lecture. And we will meet again for the next lecture. Maybe I will inform the announcement about our international guest lecture. And once again, thanks to Prof. Lehnert, and hopefully you will have a nice day, because it's already night in the US. And thanks again for your time.
Yeah, you're very welcome.
I will close this session, and Assalamualaikum warahmatullahi wabarakatuh.
Have a nice day, and I will now go relax because it's late here.
Yes,
enjoy
your night. Okay.
Goodbye.
Bye bye. Yeah.